Um, Next we have is something that's called the DNA probe. So this is something that they can use to see if a person inherited a cystic fibrosis allele. The way it works, they take the DNA, they attach it to a solid surface. Then they have this probe that's complementary to cystic fibrosis allele, and it's labeled with this fluorescence label. So it's going to light up. So they're going to put this with this DNA. And if it's able to attach it to a complementary base pair with the cystic fibrosis allele, the person has the cystic fibrosis uh, allele. If it's not able to bind, that means they're missing the complementary base pairs to this. They don't have cystic fibrosis allele. So they're healthy. People can do this for a lot of different diseases. One that's a really famous one is a BRCA gene. I shown you that before. So BRCA gene, BRCA1 gene actually. So this is the one for cancer, and I think Angelina Jolie, she got she got tested for it, and it lit up. So they probably used a DNA probe and it lit up. So she had the gene for that to get cancer, or to, she might have cancer in the future. So she went ahead and she did the preventive care ahead of time. She got rid of the gene even before it caused the disease. So this is they probably something they might have used to scan for it. So they can scan for the harmful alleles. If the person have it, then they can try to do something about it. But again, you have to be able to afford it. So it can test for mutations from any gene in the embryo, child, or adult. That's very important. Um, so if somebody gets the embryo scanned and they say your child's gonna have maybe cystic fibrosis, um, then it can change the decision for the female to have the child. She can go in depression. She can say, I don't want to have the child anymore who's already going to be sick. So that's another ethical issue. So another one we talked about is hemophilia. The royal family of England, Queen Victoria was the carrier for hemophilia. They can detect that, and sickle cell disease and Tay and disease. So they can detect few diseases. The type of question I might give you is I might give you a DNA sequence, and I say, this is the DNA sequence for cystic fibrosis allele. I'm just going to use that as an example. So I'm going to say it's AGATAAGGCT. Then I will ask you, what is the probe? What is the DNA probe for this allele? You have to remember the probe is complementary. So you're going to say it's TCTATT. It's going to be multiple choice, but you have to pick the correct one. Or I can give you the probe, and then I can say, what is the complementary base pairs for the DNA for this allele? So you're gonna have to pick the probe, but it's gonna be always A with the T and G with the C, so complementary. So there is something called PGD. I want you to remember what it stands for. It stands for pre-implantation genetic diagnosis. This is used to help reduce the odds of having a child with a genetic disease. This is something that's done in vitro IVF, in vitro fertilization. So they take man's sperm, they take few of woman's eggs, they put it in the laboratory dish, and they fertilize it with the man's sperm. And now you have few eggs that are fertilized. Then they start scanning the eggs. They take one egg, they scan it, whatever disease they're looking for. If it has the allele for it, they discard it. They take the next one. If it has the allele for it, they discard it. They take the next one. Maybe this one is healthy. Then they can take this one, put it in female's body for implantation. They can implant it. Now she can have a child who doesn't have any genetic disease. And then you have these remaining eggs. These they're trying to use for the stem cell harvesting stem cells for research. So there are a lot of people who don't want that. They don't want that happening. They don't want that happening to their eggs. So they would rather have them discarded than use them for research. So fertility clinics, they use these eggs for research sometimes, but there's a lot of ethical issues that surround that. So even though these eggs will be just thrown away, people still don't want their eggs to be used for research. Um, but if, let's say, all of the eggs had the disease, there was no egg that was healthy. Now the female might say, I don't want to have a child. I don't want any of these eggs, so this has to be redone. So they can do that. That's called pre-implantation genetic diagnosis. So researchers then extract DNA from one cell of each developing embryo. So they don't, so the way they scan for this embryo, I said they scan it, they take just one cell and look at the genetic information. Now the one cell might not have a lot of DNA, that's where they can use PCR. There's a lot of different techniques coming together. They extract the DNA first. There's a DNA extraction lab that you might have to do on the MicroHealth Connect. It doesn't work the same way. I think that's for a plant. So they extract the DNA from this egg. Now that's just, very little amount of DNA. They use the PCR, they amplify it. Then they use the DNA probe and they search for the harmful alleles. And DNA from children can be screened to see if they might have the disease in the future, or adults can be screened as well. Either they might have the disease in the future or they already have it and they want maybe something done about that. We're gonna talk about that next. What can be done about that? So that's PGD. It can identify genetic diseases. Any questions so far? Nobody really asked a question today. Any questions? Okay, so now they scanned maybe an adult or a children. Let's say they scanned somebody who have the disease. So what they're gonna do about it, there's a children. She was scanned for cystic fibrosis. The gene for that is CFTR. So cystic fibrosis happens in people with mutations in the CFTR gene. You don't have to remember the name of the gene. So it's in the lung cells. They produce abnormal CFTR protein. There's a lot of mucus. So they have a faulty gene. They're making abnormal protein. Then they can take healthy gene. They can insert it in a virus and they can have the person inhale the virus. They obviously remove the viral DNA first. They just put the healthy gene in there. The person inhales it and lung cells, they can take up the healthy gene and now they can produce healthy proteins, normal CFTR proteins. That's called gene therapy. So there is still, it's not 100% successful. And the reason for that is that your body might reject it, might not take up the gene, might not go in the correct place. Another thing is the viruses. They're using viruses. Viruses, you already know, it can cause immune response. So there was a case of a person who was trying to have a gene replaced for metabolism. They inhaled some viruses and so they gave him the healthy gene in a virus. The person inhaled the viruses, but it caused a immune response, immune reaction, very, very strong one. It stimulated immune reaction, and the person died within few days. After that, I think that was in 1999. After that, they really restricted the rules on this, so it can go wrong. So the overall goal here is to supplement a faulty gene with a normal healthy version. So they want you to have a healthy gene so you won't be sick anymore. This is just written out what's being shown here. Cystic fibrosis is caused by CFTR gene on your chromosome number 7. Don't worry about the name or the number. The result is abnormal CFTR proteins in the lung cells. They start with the virus. They infect the lung cells with the virus. They remove the viral DNA, make the recombinant virus containing healthy versions of the gene. Patient inhales the recombinant viruses, and then the recombinant viruses inject their genetic material, including the healthy gene, into the lung cells. Lung cells can now produce normal CFTR proteins. This makes it look really easy. It's not that easy. It doesn't always work like that.